Thank you so much for the introduction. I'm happy to be here to share our recent work about cognitive aging and its effect on the brain circuit. I was a previous long-term fellowship awardee, and now I'm an assistant professor at NTU in Singapore. So here's a little bit background about cognitive aging in general. So as we know, unfortunately, our mental abilities decline as we age. But interestingly, not all, but only particular aspect of the cognitive functions decline as a function of the age. So for example, if you look at the vocabulary knowledge and procedure memories, they are relatively preserved well, even in the older adults. However, in contrast, when you look at long-term memory and working memory, they are quite sensitive to aging. To see what's going on, so actually, a bunch of functional MRI studies have reported somehow the abnormal activity patterns in the older adults during some specific cognitive task. So for example, one report from the Randy Buckner's group have shown that during the semantic classification task in the young adults, if you look at the activation pattern, they showed some robust medial prefrontal cortex activity, which was significantly correlated with the posterior brain region. So this means that the prefrontal cortex are forming a specific brain-wide network, and they cooperatively work together with other brain regions to support semantic classification. In contrast, interestingly, when you look at the older adults, they showed activation, however, they're less correlated. Looks like the interregional correlation was disrupted in the older adults. Such macroscopic functional alteration may be inferred due to some morphological change in the prefrontal cortex neurons. So for example, if you pick up the prefrontal cortex neurons in the monkey dorsolateral prefrontal cortex, so as you can see here, in the older adults, they showed a significant reduction in the number of the dendritic spines, as we can see here. So as you know, the dendritic spines are some structures that receive the input, suggesting that their communication among the neurons are somehow disrupted. So this is a quite nice snapshot of the morphological change found in the medial prefrontal cortex individual neurons associated with aging. However, we have to admit still there's a substantial disparity and gap between such macroscopic functional alterations versus subcellular resolution morphological change associated with aging. So to fill this gap, we decided to study what's going on at the single cell levels and the circuit levels. So we used active behaving mouse. So we trained the mouse to perform working memory tasks as described here. So in this task, we presented bimodal stimulus, which include auditory and tactile stimulation to instruct them to remember left licking or right licking, depending on the stimulus. And after that, we provided a delay period for 2 seconds, in which there's no stimulus information there, so that the animals have to maintain and remember the left or right information in their working memory. And after that, if they lick to the correct side, they were received drop of reward. So in this study, we recruited 3 different subjects, which is young adults and middle-aged and advanced age groups, based on the Jackson's laboratory's guidance about the mouse to human age equivalents. And here we plot the learning curve in each age groups. showing different curve in different age groups. As you can see here, the young mouse showed much faster running rate compared to the other two aging groups. This is quite natural. However, it should be noted that even though two aging groups, middle age and advanced age groups, take longer training time, but still they can reach the certain behavioral criteria in the end. So after they learned this task, we performed the calcium imaging to see the activity patterns in the medial prefrontal cortex. To do so, we injected the Cre-dependent calcium indicator GCAMP6F into the medial prefrontal cortex of the CAMK2 alpha Cre mice. In combination with green lens implantation and endoscope attachment, so that the only excited neurons expressing the GCAMP6 showed some fluctuation of the intensity of the fluorescence, as you can see here, whenever they fire action potential. So this technology allowed us to measure and monitor the neural activity of more than 100 excited neurons at the same time of the mouse behavior. So here I'm showing the example of individual neurons of the activity. So this is a time course of the specific neurons and averaged across many trials. As you can see here, this neuron showed left selective activity in the sense that they are more active in the left trials than the right trials. And their selectivity was maintained throughout the delay period, as shown here. This neuron showed, on the other hand, showed right selective activity. So let me call this type of neuron as working memory neurons, because their selective activity of the left or right should be important for maintaining the left-right information in working memory. And these working memory neurons are not necessarily exclusive to the young adults, but instead, we actually identify the similar working memory neurons in the middle age groups as well as advanced age groups. But this is just a cherry-picking example, and the critical question here to ask is that how about their total number, and how about their signal quality and reliability? So here we plot the fraction of the working memory neurons encountered of the old image sessions in each age groups, young, middle age, and advanced age groups. So this means that how frequently we encounter the working memory neurons of all the imaged neurons. And as you can see here, we found a significant reduction of the fraction of the working memory neurons as a function of the age. We also evaluated the reliability of individual neurons' signals. So this is a selectivity index, which means that, so we evaluated how reliably the neurons tell left or right information during delay period. So the higher value means that the neurons reliably fired consistently across trials, either left or right trials. So as you can see here, again, there's a significant reduction of the left or right selectivity as a function of the age. So it looks like both quantity and quality-wise, the working memory signals dropped as a function of the age. So as a consequence, when we did a decoding analysis by using the population of the activity recorded in each age groups, so this is a time course of the decoding performance, which means that we can predict the left or right licking response by taking a look at the population activity at each moment. And so 100 means that we perfectly predict the left licking or right licking by taking a look at the population activity at each moment. So as you can see here, the young adult population activity perfectly predict left or right flicking response, especially during delay period. But their decoding performance significantly declined as a function of the age. So this means that the working memory information maintained in the population of the medial prefrontal cortex decline as a function of the age. So we further wanted to understand what is the circuit mechanism underlying such working memory loss. So computational modeling work suggests that specific activity patterns can be stably maintained with the recurrently connected networks. So the recurrent networks should underlie the working memory maintenance circuit. So here we have two specific hypotheses. Number one is that the working memory neurons we identify should have stronger mutual connectivity among the working memory neurons. And then they should form our recurrent networks, which can stably maintain the left or right information in their recurrent networks. And number two is that these recurrent networks formed by the working memory neurons should decay and should degrade as they age. To test this hypothesis, we measured the spontaneous activity from the mouse in the same brain regions, but now outside the task context. So the mouse are just sitting there, but not doing any behavior performance, just for 10 minutes. So this spontaneous calcium imaging allowed us to evaluate how each pair of the neurons showed correlated activity among each other. But by taking a look at the same population number with task or without any task, so that we can evaluate and quantify the functional connectivity strengths among each pair of the neurons. So here we plot the functional connectivity. First of all, this is the data set of the young adults, and this is an irrelevant pair functional connectivity as a function of the physical distance among them. So this means that, so irrelevant pair means the working memory pairs versus non-task related neurons. As you can see here, the functional connectivity decays as a function of the physical distance. This means that so nearby neurons tend to fire together. However, nearby neurons tend to fire very strongly. However, the faraway neurons are less likely to be correlated, which is a natural phenomenon. So the question is, how about the working memory neuron pairs? Here I show the strengths of the functional connectivity among working memory neurons. As we predicted, they form a strong mutual connectivity among them compared to the irrelevant pairs. Interestingly, if you look at the data in the middle age groups, they show significant reduction of the functional connectivity, especially between the working memory neuron pairs. And more interestingly, if you go to advanced age groups, there's a further reduction of the functional connectivity. And importantly, if you look at the difference between the working memory neuron pairs versus irrelevant pairs, we can see a significant reduction as a function of the age. So this means that, so this supports our second hypothesis that the working memory neuron pairs recurrent networks already decayed, and this is starting already at the middle age stage. So this result further predicted that the middle age groups, the functional circuit in the working memory in the medial prefrontal cortex may show more vulnerability to any perturbation. To test this, we used optogenetic techniques. So we injected inhibitory opsin called GTACR2 expressed in the excited neurons of the same medial prefrontal cortex. This technique allowed us to temporarily and reversibly inhibit the action potential of the target region by providing the blue laser like this, so that the action potentials are totally gone after the laser stimulation. And if the laser is off, the action potentials are recovered soon. So in this experiment, we provided only brief duration. So we targeted early half of the delay period to give a mild perturbation of the circuit to see what happens in the behavior. So this is a result of the behavior in young adults and middle-aged groups. So as you can see here in this laser off condition, so the middle-aged groups showed quite comparable working memory performance as the young adults. So which means that without any laser stimulation, they showed quite normal working memory performance. However, the mild perturbation caused more severe behavioral impairment compared to the young adults. So this means that middle-aged groups showed quite normal working memory performance at appearance. However, behind this, their working memory circuit are already start to show vulnerability to the mild perturbation. Also, with that, I would like to thank all my laboratory members for their significant contribution to this work, and thank you so much for your attention.